to kick off this day, you can already applaud him. I mean, to be the first on stage is not that easy. Marwan and Chazli from Myriad Optics. Marwan.
Thank you. Imagine it's Friday night. You are at a fun party with your friends and you're having pizza. The next day you all wake up at the hospital because the pizza was contaminated with bacteria. Now this is not fiction, and it famously happened a couple of years ago. And actually every single week, products that you and I can buy on supermarket shelves are getting recalled because they're found to be contaminated with deadly bacteria. Those cause tens of millions of tons of food waste every year and cost billions of francs for the food companies. Why is that? Well, suppose you're a quality control manager at a food factory. You want to check for listeria on your sandwich production line. You start by taking a swab sample on your line and you send it to a lab where they're going to put your sample in something that looks like this. This is a petri dish. In here, the bacteria are going to grow and multiply until there are literally billions of them, and then and only then can you actually test them and know if they're dangerous for your customers or not. The problem is bacterial culture is extremely slow and can take up to 5 days before you get any results, and by that time, your product is already in the hands of your customers. This is where we come in. My name is Marwan Chazli. I'm the co-founder and CEO of Myriad Optics, and with 3 EPFL engineers, a microbiologist, and along with brilliant advisors, we are on a mission to transform the way that quality control is done in the food industry today. We're developing a solution that will take the time you need to detect bacteria from several days down to just 1 to 4 hours, and all of that right next to your production line. We're achieving this by combining a microfluidic chip, which can collect and trap the very small numbers of bacteria in your samples, with Raman spectroscopy, a laser-based optical spectroscopy method which can scan and identify single bacterial cells. Our product will consist of a swab to collect your samples, the microfluidic chip, and a tabletop device that can rapidly analyze those samples. And we're currently in the process of patenting our technology through EPFL. Our primary market is the food industry, with a current size of over 3 billion francs. And we've interviewed customers in dozens of companies, including PepsiCo, Mars, Nestle, Bühler. And we've secured 2 letters of intent, including pilot test commitments with Migros Industry and Iceberg. Up until now, we have raised just over 430,000 francs, and we're well on our way to reaching our first prototype. And with your support, we could go even faster and make sure that everybody stays safe next time we all have a pizza party together. Thank you.
Thank you very much. Yeah, it's tempting to clap, right? Thank you so much. Who would like to start for the Q&A? Christian.
I will start. Very good presentation. Thank you.
Thank you.
I have a question to the innovation level. You use standard technology. So what is really unique at this approach? And do you have a patent filed or what?
Absolutely. So it's true that Raman spectroscopy and microfluidics are technologies that are known and used commercially. Where we're innovating is in the design of our microfluidic chip, which allows us to trap the very small numbers of bacteria in industrial samples. So we're able to break the sensitivity limit. And the other one is in the proprietary buffer reagent that we're using. And we're currently in the process of filing the patent with the EPFL. We haven't obtained or filed the patent yet, but we're working with a patent agent to draft the claims at the moment.
What are the expected costs for this chip product?
Yeah, so our first pricing, we're aiming it for 25 francs a unit, which is something that is comparable at the sale price with a production price that is about half of that, which is comparable to other existing solutions of that type in the market.
Thank you. So Teresa getting ready. Teresa?
Thanks so much for the presentation. Can you tell us a bit about the competition? It seems really like a quantum leap forward. And do you have anybody at all close to where you are?
Yeah.
Beyond that, I'd like to understand a bit more about the readiness of the market to adopt this?
Yeah, absolutely. So we have two categories of competitors. The first one is the traditional methods. So that's the Petri dish-based methods that are then used with PCR or MALDI-TOF. And this is currently the standard that we need to replace if we want to penetrate the market. There are several startups that are working on faster detection. And none of them are taking the approach exactly that we're taking. And the fact that we have this combination of technology allows us to have USPs that are more interesting than the existing competitors. But it is a space that is very interesting for a lot of people. And in terms of the readiness, for us, we want to drive the appetite of people in the food industry by the USPs that we provide. So we're telling them, instead of getting a result after 2 or 3 days, you get it within 4 hours. And if they get it after 3 days and they have a positive, that means everything they did in that period of time goes to the trash. Whereas if you get it within 1 to 4 hours, then you save a lot of money, time, and product.
Thank you very much, Marwan.
Thank you.
Do you mind taking the floor? Do you take the table? Next to come on stage for us is Edwige Guinet for Visienco. Thank you, Edwige.
Today, in the drug development process, over 90% of drugs tested on animals fail to work in humans. And this is why pharma companies have invested in a new alternative, organoids. Tiny versions of human organs grown in the lab, just a few millimeters in size. So just imagine a mini brain or a mini liver. But organoids are going beyond reducing our reliance on animal testing. They are revolutionizing medicine, helping scientists develop better drugs and personalized treatments. Now imagine you develop a disease. Instead of going through a series of potential treatments, your doctors could test drugs directly on the mini versions of your own organs in a non-invasive way to find the best therapy for you. Sounds really promising, right? But to achieve this vision, there is a problem to solve. Organoids are hard to scale. Today, in labs, technicians still sort, transfer, and analyze organoids one by one manually. This slow and costly process is also prone to human mistakes. At Visienco, we solve this issue with the Orgadroid, a compact AI-powered lab automation device which automates these manual steps. It boosts throughput by a factor 7 and increases reproducibility to 95%. What makes the Orgadroid unique is its ability to sort large organoids in a gentle way, ensuring viability and at scale. The technology, coming from Swiss Research Center CSCM, is protected by 4 exclusive patents. And the impact of this technology is clear. More efficient labs, reduced reliance on animal testing, life-saving treatments reaching patients faster. And our solution is already making a difference today. We recently completed 2 pilots, including one with pharma leader Roche. We secured our first sales with lab automation provider Zeiss. And we are now in the process of building a strong sales pipeline with pharma, biotech, and research labs. And we have 3 LOIs recently signed. To continue on this journey, we're now raising a seed round to commercially launch our product in Europe this year, prepare for our launch to the US market by end of 2026, and continue developing products and services to further optimize the organoid production. We are Visienco. My name is Edwige Guinet, and I'm CEO and co-founder. Organoids are revolutionizing medicine, and at Visienco, we make this revolution possible. Thank you very much.
Merci. Merci beaucoup, Edwige. Let's change the rule. Let's clap straight after the pitch. It feels way more natural. You're right. Who would like to start with our Q&A?
Thank you very much. Very interesting story. I would be interested in the pilot you have with Roche. Can you tell me a little bit more what they're doing there?
So I can't go into too many details, but basically we had 2 use cases about 2 types of different organoids. So brain organoids on one side and retinal organoids on the other side. So rather large organoids, so really typically a few millimeters in size. And the idea was really to help identify within these 2 pilots for our clients or users, let's say, the ability for the system to to transfer the organoid first, but more importantly, classify organoids and be able to identify, for example, the retinol organoids that we would keep in the following of the process so that we could start testing compounds on them. So typically for retinol organoids, we were looking for specific shape, form, specific elements so that our AI model could identify them and sort them accordingly.
Christian, and then Felix.
Could you elaborate a little bit on the business model and the market potential of this technology?
Sure. So the organoid market is planned to reach 15 billion by 2031. And in terms of, we address the lab automation market, so it's about 30% of that. So it's about 5 billion by 2031. And in terms of our entry points, we're looking at entering through the large organoid market, which is really our strength, where our USP lies. And in terms of business models, so we do direct sales to pharma, research labs, and biotech companies, but we also, with ZEISS, we're working on OEM integration of our device. So basically, they are building their own production line of organoids, and they will integrate our organoid as part of their own production line. So every time they will sell a production line, they will sell one of our devices. And typically, we are looking also at having more collaboration like this one.
Felix?
Sounds really interesting, but you have 4 patents.
Yes.
Are you selling a machine? Are you selling software?
Yep.
Are you selling services? What is the patent and where do you make money?
Okay, so there are 2 questions here. So the first and most important patent is related to our tip that has been specifically designed to sort and transfer large organoids, so it has a specific shape. Another patent is related to the organization of the device. It's a bit technical, but happy to discuss this more in detail. And the 2 other patents are related to a redistribution system that we have as well. In terms of what we sell, we have so the device with a basic pricing of 100K. On top of that, we have a custom AI model, 20K. And from there, we also have consumables with this tip that I've mentioned, patented tip, maintenance, but also a software solution that we're defining right now. So from the different use case and conversations we have with players on the market, we see that there is a need for software and for analytics with regards to the organoids that are being sorted. There is strong need, so that's what we're developing on top. Yeah, and that would be licensing.
I'm sorry, we have to move on. She'll be there at the break.
I'll be around, yes.
Thank you very much, Edwige.
Thank you.
Our third startup for the Life Sciences, Biotech and Pharma cluster is Estelle Clerc for CellX Solutions. Estelle?
Hi, everybody. I'm Estelle. I'm the CEO and co-founder of CellX Biosolutions, which is our startup that is based on my PhD research at ETH Zurich. So at CellX, we're tackling one of the most pressing environmental challenges of the century, which is the sustainable treatment of chemical pollution. Because we know that every year more than 350 million tons of hazardous chemical waste is produced in industry. So think about byproducts for pharmaceuticals, pesticides, microplastics, and many more. And that's a real problem, because the methods to treat chemical waste, they're really not up to the task. On one side, we have traditional methods, which is, let's face it, incineration of this chemical waste. It's really expensive and not sustainable at all, while on the other side, bio-based methods are emerging, but they really remain early stage, because so far, we haven't had the most performant organisms. Therefore, there's no commercial solution at scale on the market. So at CellX, we are here to revolutionize bioremediation by creating an on-demand catalog of highly performant bacteria that can degrade pollutants efficiently. Think of us as the Netflix of bacteria. You can pick and choose based on your needs and wishes of biodegradation. So how does this work? How have we been doing this? So during my PhD, I developed a microfluidic device that acts as a selective screen that allows us to capture in the environment, like here in the ocean, bacteria that have natural abilities to degrade pollutants. And we pair it with robotics to sample in any type of environment. After they capture, we bring them back into the lab and we turn them into a product that will be then implemented in the environment or in industry to produce clean wastewater. So far, happily, we have already assembled a library of more than 50 bacteria prototypes able to degrade pharmaceuticals, pesticides, microplastics, and the infamous forever chemicals, PFAS. So we've calculated that for a medium-sized chemical company producing about 50,000 tons of chemical waste per year, using our biosolutions would reduce the treatment cost by 5 times compared to incineration, but also avoid CO2 emissions and dramatically reduce the impact of these chemicals on our health and on our environment. So far, we have already 1 paying client, 2 pilots are in negotiation. On top of our bacteria prototype library, we have 1 patent pending for the tech, and we have obtained 1.4 million in grants already last year. And I'm really happy to be able to tell you today that yesterday we closed our pre-seed round for 1.5 million. So I'm really happy to be able to count on your support today to step together towards a greener future. Thank you very much.
Thank you, Estelle. Ladies first. Teresa?
Thank you, Estelle. Can you comment for us on the scalability, the industry application? I assume that you've done most of this on a laboratory scale until now, and also any regulatory issues across geographies.
Yes. So now we're at a lab scale, for sure. We've already scaled up about 100 times from microfluidic size to culture tube size. But then we have access to bioreactors, and this is what we're going to do in the next months to really look at the stability and reproducibility of the bacterial function in half a liter. And then we have already found some partners to be able to go 100 times, up to 110 to 100 liters. This is where we're going to be able to determine product formulation before going into manufacturing. Regarding the regulations, so basically we have a big advantage here because we're using natural bacteria. So they are not been modified. This is our big advantage compared to GMOs or enzymes. And because we have taken them from the environment, we can also easily put them in the environment.
Thank you. Felix.
Are you selling the bacteria, or are you selling a software or machine? And how long is it going to take? I have a big field and there's a lot of PFAS in it until I can call you and you deliver me the bacteria to get rid of them.
Okay, so how long is it going to take? So now our PFAS degraders, so PFAS are the forever chemicals that are not supposed to be biodegradable. These are our most advanced products, actually. We are able to degrade at the moment more than 50% in 7 days, which is by far, as much as we know, the most advanced product that there is. And we expect that we can basically go on the market at the end of 26.
Yes, we have time for one more.
But I have difficulty in understanding. On one side, you say they are natural bacteria. On the other side, we say they are forever chemicals. So why these things don't match in nature?
So this is a very good question. So I think, bacteria are so diverse and they have so amazing metabolic abilities. We just need to be able to find the right ones. And this is exactly what we're able to do with our microfluidic device. We're able to find a needle in the haystack. And so far, we're not the first ones showing that we can degrade them. There are a couple papers in academia that show that they have also strains, just 1 or 2 strains that show also the degradation. So it's more that we're now transforming these cocktails of bacteria that we're assembling at a commercial level to bring it to the market.
So they are applied in the sewage systems, then, these bacteria, or where you release them?
So the first implementation spot is going to be in industry, where industries, for example, PFAS producers, they store their PFAS byproducts into reservoirs, concentrated waste, before sending to incineration. And this is where we can basically install our bacteria into a bioreactor to get rid of these chemicals. And then non-toxic wastewater would end up into the wastewater treatment plants. But the end goal is really to be able to do bioremediation in situ, in polluted environments, and this is the biggest market.
Thank you. Thank you very much, Estelle.
Thank you.
The next entrepreneur stepping on stage is Reiter from Teno Bioworks. Willkommen.
Good morning. It's an honor to be here. I'm Michael, co-founder and CEO of Teno Bioworks. Over the past decade, I've been working on reducing the planetary footprint of chemicals, because chemicals really form the invisible backbone of modern life. Your smartphone, your clothing, the materials in our building around us all contain them. However, today, most of them are still being produced from fossil resources. This emits 6 billion tons of CO2, making chemicals one of the major drivers of climate change. We can also make chemicals from sugar derived from agriculture. This saves some emissions. However, this comes at the expense of massive water and land usage, as well as negative biodiversity impacts. Additionally, using sugar for chemical production competes with our food supply and ultimately also doesn't scale. At Teno, our mission is to overcome these problems and make chemicals that are planet compatible. To achieve this, we start with a novel starting material called green methanol produced from CO2 or methane. We convert this very simple substance into complex chemicals using E. coli bacteria, a technology breakthrough several years in the making at ETH Zurich. Think of E. coli as microscopic factories that we can now program to turn methanol into chemicals of choice. The result? Carbon-negative chemicals that also protect ecosystems and biodiversity by using 15 times less land and 100-fold less water than sugar-based chemicals. Together, these benefits make our chemicals truly planet compatible. I'm sorry. As first products for our platform technology, we're pursuing shikimic acid and aminobenzoic acid. Both molecules have large, growing, multi-billion-dollar markets and applications across an array of different industries. For example, you may be familiar with Tamiflu, the drug pioneered by Roche, is produced from shikimic acid. Last year, we set the foundations of Teno Bioworks. We won the Bridge Discovery Grant, demonstrated proof of principle production as well as lab scale-up of our technology, filed patents, and started engaging with industry where we're seeing strong interest by leading chemical, cosmetics, and pharmaceutical companies. This year, we'll scale our technology, sign letters of intent, and raise a pre-seed round to accelerate scale-up. All of this is driven by a team of 3 co-founders, blending engineering excellence and entrepreneurial spirit. So, to end, we believe we've unlocked a new path for the scalable, cost-competitive, and planet-compatible production of chemicals. If you'd like to join in on this vision or see opportunities for collaboration, let's connect. Thank you.
Thank you, Michael.
I think this has come off now.
That's all right.
Okay.
Who would like to ask? Christian, yes.
Thank you for the presentation. What exactly do you sell?
Yeah, so our business model will be that we become a technology provider that develops both the bacterial strain as well as the overall production process. And once we have proven sufficient maturity of our technology, we'll license it out to large chemical and other companies for global scale production lines then.
Okay, so first is a service for develop the exact bacteria, and then it's licensing.
Exactly, and it's bacteria plus the process because both things have to come together to really make it work.
Thank you. Wolfgang?
Thank you for your presentation. Looking to your application, you worked as scientist the last year. What is the greatest challenge for you to become an entrepreneur?
So I guess one of the challenges in general is, I think that it really goes for a lot of entrepreneurs, is that going from like the technology that you developed to solving real world problems. And this really, I think, took now some time to wrap my head around and really think about the problems rather than, oh, look, I have this technology, especially with our platform technology now that we can use for all sorts of things. Okay, what are really the best problems that we can start solving now?
CellX.
Why is your platform going to succeed and not others?
So yeah, for methanol to, or green methanol to products, there are different approaches. There are purely chemistry-based solutions, and some of them already work pretty well, and we will not compete with those technologies because we use different target molecules that we want to produce. There are also others that want to use bacteria, and here we see the big advantage that we have is that we're using E. coli bacteria, which are already established in the industry. Other people use organisms that are out there in nature, but we don't know how to grow them at scale. If you put them into a large industrial plant, they stop performing the way they performed in the lab. And this is really where we see our big advantage with using a very much industrial established organism.
Thank you very much, Michael.
Okay, thank you.
Thank you. The next lady to come on stage is Chloé Albietz from Bioscibex. Welcome.
Thank you for your time. My name is Chloé Albietz and I'm the CEO and co-founder of Bioscibex. Biological drugs are very effective drugs in treating cancer or autoimmune diseases, but the production process is super complex, involving 7 consecutive steps. As a result, biopharma companies lose significant amount of money every year. My co-founder, François Caruso, who spent several years working in the biopharma industry, has been obsessed in finding ways to simplify this process. And it looks like he was not the only one, because we interviewed many different biopharma companies, and they're all looking for a solution to this problem. Some of them are considering developing an internal solution. Others have invested millions in a robotic platform, too complex to be used. Others are dreaming of an equipment that would simplify the process and take out some steps. And we want their dream come true. That's why we're building Bioscibex. Our bioreactor is enabled to decrease the number of steps from 7 to just 2. We will save time, space, product loss, and downtime, and we'll save manufacturing cost. The origin of the idea is actually inspired from the Wave bioreactor that was patented in 1998. Wave had a brilliant idea, but it was so disruptive that at that time nobody believed it could work, and it took them 15 years before being adopted and becoming a standard in the biopharma industry. We are building on this technology. We have the same vision on the bag, the same vision on the wave motion, but we add an additional axis of motion, a vertical motion. And with that, it enables us to precisely control the cell culture condition from 30 milliliter to 30 liter in just one equipment. So we are currently actually, we have a beta prototype, and we are planning to do a pilot trial with a top 10 biopharma company by the end of the year. We are a team of 2 co-founders, both with industry experience, and we are helped by talented advisors with a lot of experience in the biotech industry and fundraising industry. We are currently actually raising a pre-seed round, so if you have any interest, please come and talk to me. Thank you.
Thank you very much, Chloé. Who would like to start? Michael. They're fighting for the microphone.
Yes, we do. Thank you very much for the presentation. What is your main challenge you are facing right now?
we need to build a robust product in terms of, like, for example, the motors itself. It doesn't seem a challenge, but we have to make a really robust product, because Wave, they had similar issue in the past, and customer didn't purchase again the Wave and they'd switch. So we know that's been a concern, and we are like in a very conservative industry, so we need to make it right first time, reliable and robust.
Christian, do you say Guy?
Okay. You said that you achieve from some milliliters to liters in the lab pre-production. What is the target amount for the production you need? I suppose to make this really scalable, you need to go to several hundred liters, if not tons per hour. Or what's your target there?
So the biologic production, even though if you want to produce 10,000 liters at the end, which is like a standard, let's say between 2,000 and 20,000 liters, you always start from 30 milliliter. So you always start from a very low amount of cells that you multiply, and that's why you have all those steps. We are developing a solution for all the early process, and then with our solution, you would have just one equipment and a second one, and you have the production done, basically. So it's a solution that's for the production scale, because you always start at 30, even at production. So it's not a scale-up, it's not a lab only, it will be used also for manufacturing purposes.
Thank you very much, Chloé. Time is up, so I think we're going to stop here. Thank you again, Chloé. Our next startup on stage is José Luis Bila from Precise Health. Welcome, José.
So, hello everyone. My name is José, and I'm the CEO and co-founder of Precise Health. I lost both of my parents to antibiotic resistance. In one case, the doctors did not have the time to act with the right treatment. In another case, they simply did not have the right, they didn't know which other antibiotic they would be able to prescribe to my parents. So the sad fact is that my parents were not the only ones. And by 2050, it is estimated that there will be more than 3.4 million deaths from antibiotic resistance only in time-sensitive infections. To make matters worse, multi-drug resistant infections already today are killing at rates higher than 50%, not to mention the economic impact that is having. Now, among the plethora of solutions that have been proposed today, bacteriophages are by far the best alternatives to antibiotic-resistant infections. And bacteriophages today, so for those who do not know what bacteriophages are maybe, are tiny microorganisms able to selectively infect and kill harmful bacteria while leaving the beneficial bacteria alone. Today, bacteriophages are used in compassionate use under magistral applications. However, there are trillions and trillions of bacteriophages in nature. So finding the right one takes up to 8 weeks. So this model simply is not compatible with time-sensitive and multi-drug resistant infections. At Precise Health, what we are doing is developing a platform using a combination of genomics, data science, and machine learning to fast-track the treatment of multi-drug resistant and time-sensitive infections using machine learning in this case. And to pinpoint the best bacteriophage, or to improve the success rate of finding the right bacteriophage, Precise Health is integrating a multitude of data from multiple biobanks around the world. So the doctor can just upload a genomic information into our platform of the bacteria, and it will show which bacteriophage is working in any biobank that is integrated into our solution. So we are learning from one platform, which is the phage match, which is the matching platform, and these learnings will be integrated into a more advanced model, which will be able to engineer in silico de novo bacteriophages altogether. Now, to validate this, we have developed a proof of concept pipeline. And we are currently working on bacterial sepsis, and we are already advancing into animal studies as of Q2 2025. This is just a snapshot of what the matched results look like compared to current broad-spectrum antibiotics, which you can see from the blue line that our bacteriophages are much, much better. Now, to make this work, we have a team of experts from all different fields that complement each other, and they are all working under one roof under one simple mission, which is combating AMR one bacteria at a time. So, thank you. Yeah.
Thank you. Thank you very much, José.
Yeah.
Teresa?
Thanks so much. Can you comment on the immune response to the phages in the patients?
So far, the immune response, it's really dependent on the dosing that you're using. Sometimes, if they are too potent, they are acting too fast, and this can cause inflammation, so you need to give anti-inflammatory as a counter-response to that. So finding the right dosage is key to that. But so far, the ones that have found the right dosage actually did not have many side effects. So actually, they have been proven to work quite well. Yeah.
Thank you. Felix?
What differentiates your platform? Because you do AI-based antibiotics engineering using biodatabases. So what differentiates your process? Why cannot other people do the same as you do?
Good question. So the other platforms which are existing today are doing this using biodata, but it's mostly for broad-spectrum antibiotics. We are shifting the model from the broad-spectrum antibiotics to using these bacteriophages. In the bacteriophage side, currently, there is no other company that we know of that is doing this. It's mostly on the academic level that everything is being done. And I can tell you, it's very complex. We've been working on this for the past 3 years. It's really, really complex, but we found a way. So I think that's the differentiation.
Do we have one more question? Yes, Guy?
Yeah, I'd like to ask a question on your financials.
Yeah.
How do you fund this company? What do you live off? How this works?
Yeah, so maybe a good thing that was mentioned earlier, we made some financial sacrifice, personal ones, which we've been doing so. We have had some grants up to 300,000 plus angel investments, which have complemented. This is how we have done. So with very little money, we have managed to accomplish a lot, but it takes a lot of financial sacrifice. This is where we are, and this is why I think the money from the foundation would be very, very helpful to take us to the next step.
Thank you very much, José. I promised I would say his name right, but I do not speak Italian. Gianvito Grasso from Virtuo Laboratories.
Hello, everybody. My name is Gianvito Grasso, and I'm the founder and CEO of Virtuo Labs. Today, there is more than 400 million people globally affected by rare diseases. It's more than cancer and Alzheimer's disease combined, and almost no one is getting a treatment. This is the stark reality we face today. And why is this happening? Well, it's because creating new medicine is incredibly hard. It takes more than 2 billion Swiss francs and 12 years to bring a single drug to market. But what if we could test billions of potential drugs in just a few days? What if we could find the right drug faster and cheaper? That's what we can do in our company. We have developed a computational discovery platform that combines generative AI and molecular simulations. Our technology is the result of tens of years of research in this field, where we integrate award-winning machine learning models for drug design. We address the most important point and the most important reason why the impact of artificial intelligence in this field has been relatively limited so far, which is the scarcity and low quality of data. We can train our model not only on experimental tests, but also on synthetic data that we can generate using physics-based molecular simulations. Instead of physically testing thousands of compounds in a wet laboratory, we can virtually test billions of them. Our technology can predict which molecules are likely to work, and more importantly, which one will be safe. Thanks to our solution, we can increase the success rate of drug design by more than 100%, and this will make it feasible to address neglected diseases that affect over 400 million people globally. Our business model is focused on the development of novel medicine, which allows us to retain IP to be out-licensed after the preclinical phase. This usually results in down payment, milestone payment, and royalties. My background is in biomedical engineering with a PhD in computational science, and I've been doing research in this field for the last 10 years. Our team brings together machine learning researchers and experts coming from pharma companies with more than 30 years of expertise. We already started paid projects in collaboration with pharma companies, and we have just closed our first financing round of 2.7 million Swiss francs to build our technological infrastructure and to advance our proprietary drug discovery projects focused on neglected diseases. That's why I'm here asking for your support to accelerate the development of life-saving medications. Thank you.
Thank you very much. They are looking at each other. Who is going to go first? Felix, yes?
You have 100% higher success rate, you said. So creating a new pharma product is 1 out of 1 million or 1 out of 1 billion. 100% better is 2 out of 1 billion. Is this good enough?
This is a good point. So the process, the drug discovery process, is extremely risky. Fortunately, the percentage are a little bit higher in the sense that in the discovery phase, the probability of getting a drug is around 30% with this kind of technologies. And when I said the probability is 100% higher, of course, it's an assumption because we still don't have the clinical validation, right? And it's an assumption based on data because the AI discovered molecules already today, they have 100% higher success rate. And as you can imagine, they are in the clinical phases right now. So it means they've been developed like 10 years ago with extremely, let's say, outdated machine learning models. So of course, the only point that actually matters is the clinical success. We have state-of-the-art models, so I hope we will be able to demonstrate this as soon as possible.
Thank you. Christian?
What is the time benefit with your process? I think this is important factor.
This is also important. Our methods impacts the time, in particular in the discovery phase. And the problem is that the discovery phase is also a kind of smaller percentage of the whole time, which is extremely related with the clinical phases. So of course, the discovery phase is faster. And I will say it's important that machine learning models allows you to fail faster. This is also related with the reason why then the success rate is higher.
First target is?
We have 4 targets. The most advanced one, the first computational discovery platform that we are building right now is focused on nuclear receptors. They are the reason of more than 100 pathologies. Most of them are rare diseases, and this is what we will deliver within the next few weeks and months.
Thank you very much, Gianvito.
Thank you very much.
We're staying with Biotech with Freddy Hunziker von Neurods. Willkommen.
Hello, everyone. It's a pleasure to be here today. Today, I want to show you how an amazing raw material with 40% of proteins and years of research can disrupt and revolutionize the entire dairy industry. About 10 years ago, I had a bigger accident. I turned vegan, I missed cheese, and I started to found New Roots together with my partner, Alice. With New Roots, we want to craft the traditions of tomorrow by replacing dairy with plants. Our main mission is to work with Swiss farmers, cheesemakers, in order to bring a new raw material into the Swiss or European agriculture to change the entire dairy landscape from dairy to plants. The reason why New Roots is successful is mainly only because since the beginning we have been doing everything in-house. whether it's engineering or processes, building or infrastructure, researching and developing, and also doing our own agricultural monitoring. As of today, we're a team of 40 brilliant minds with a strong focus on research and development, and we have just won the award for the most innovative company in Switzerland. What is true innovation? From my perspective, innovation is a product which is not solving only one aspect or one problem. For me, innovation is a product that is solving many, many problems along of one of the most important industry, the food industry. And let me show you our solution. With years of researches, we have breeded a new raw material with 40% of protein. We are mixing that up with local, fresh Swiss water. And then we are addressing our patent-pending technology using precision fermentation in order to break down proteins, fats, in order to have a product which 100% deserves the name cheese, but without any downside. Innovation always depends on a certain timeline. It was very innovative about 3,000 years ago to keep a cow, take its milk, and produce cheese. As of today, we have total other circumstances. Not only is it obsolete, but it's also highly inefficient. With our newest technology, first of all, we can produce dairy without subsidies and about 40% cheaper than actual dairy, because we don't need to go through the middleman, the cow. It takes about 4 years that a cow can produce milk. Thanks a lot.
Thank you, Freddy. Right on time. Christian.
Of course, I read your case, so you use lupins to create that raw material. You didn't say it. I'm interested to understand what is the overall balance, because you need big fields, you need chemicals to produce it. Is this not a bit the downside of this innovation?
It's a very good question. Actually, we looked at the dairy industry and started to realize how can we solve many aspects. And the lupin actually doesn't need chemical. It brings back nitrogen into the soil in order to regenerate the soil. Very, very crucial, okay? So we can use it as a crop cycle. And again, it has 40% protein. So in just 3 months, we can generate on 1 acre of land about 1.5 to 1.8 tons of protein. So any bioreactor will never be as efficient as the lupin or as with that system.
Felix?
When can I taste your cheese? Will it taste okay? And is it healthy?
So exactly. We also try to look at dairy and started to realize that everybody loves it, okay? It's deeply rooted in our tradition, but it has downside. It has cholesterol, saturated fats, lactose, and all of this we remove, but by keeping the taste, the texture in place. So yes, it tastes fantastic. We have just launched it at the Biofach as a new world innovation, and it had excellent feedback. So I will send you a parcel as soon.
Try it now?
Unfortunately not, because I just had 5 minutes. So next time if I have more time, we can lie up a lot of cheese wheels here.
Thank you. Teresa?
Thanks so much. The company has already proven itself. You have a number of employees. You've won a number of prestigious awards. This is an award for entrepreneurship. What does it mean to you?
I mean, what's crucial to say is New Roots is not a venture case. So we really want to do business for good. So we give, for example, 1% of our turnover to farmers who we can help transform towards a plant-based agriculture system. And it's very hard to find funds as a company like New Roots that spends around 2 million in research and development. So that, I think, is really true entrepreneurship to really maintain a social balance between economics and the impact we're generating as a business.
One last key.
Yes, thanks. You face a pretty stiff lobbying competition. How you deal with that? Because the milk lobby in Switzerland is very strong. If I look at the agricultural lobby worldwide with the borders, it's also quite tough. How you go about that?
So actually, when we started, we had death threats and everything, but we started to understand that the lobby is mainly supported by farmers, and we want to work hand in hand with farmers. We want to generate a new income, a sustainable income without subsidies in order to help them to thrive. And that's our main impact with New Roots, to get along out of the lobby work. And I think we have succeeded quite well now after 7-8 years in business with that kind of approach.
Thank you very much, Freddy. Thank you. We are getting to the end of this cluster. I would like to welcome on stage now Gian, Nutal, Schädli for CompagOs. Thank you.
Hello, everyone. Meet Sarah, a 55-year-old mother who was diagnosed with stage 3 lung cancer just 3 months ago. The good news? She fought and successfully completed her therapy. But what does that mean to be a cancer survivor? Every few months, she goes back to the hospital for checkups. There's a major hidden risk. Within 6 months, the cancer could silently spread to the bones. It's called bone metastasis. It weakens her skeleton, making it fragile, so that within less than 2 years, she could suffer a fracture that doubles her risk of death. Every year, more than 16,000 cancer patients in Switzerland face skeletal complications, and more than 2,400 of them suffer from pain and fractures that rob them of the precious time they have with their loved ones. And the only thing doctors have to predict the fracture, pain. But by the time Sarah feels severe bone pain, it's too late for an intervention to make a real impact. At CompagOs, we are changing this. We are developing a blood test to help doctors detect skeletal complications 6 months earlier, before fracture, before pain. Our goal is to give doctors time to act and protect patients like Sarah through earlier intervention from devastating effects of cancer in bone. Our team is built for this challenge. We are combining expertise and experience from engineering, automation, bone biology, science, business and medical know-how to bring our solution to patients. Our magic sauce is to replicate the biology of bone degradation outside the patient. We 3D bioprint mineralized living bone models complete with stem cells. We call them bonoid. Then we train them with cyclic mechanical loading to enhance mineralization so much that they turn white just like real bone. Then we introduce from the cancer patient blood cells, specifically those that are manipulated by cancer to resorb bone, and we test how aggressively they cause fractures in our bonoids. This lets us assess fracture risk and guide preventive care. I'm committed to this journey. In 2021, I finished my PhD and I shared with my supervisor my dream of making a difference through a startup. This led to a postdoc grant, applications in rare bone disease and bone cancer. We created hundreds of bonoids, and in 2023, we incorporated CompagOs. Since then, we kept refining our technology based on clinical input and have now 4 LOIs from hospitals that want to work with us to bring this solution to patients. This year, we start with pilot testing with the Canton Hospital from Fribourg for lung cancer patients and work on automation with CSM. Your support helps us to achieve key milestones faster. Thank you.
Thank you very much. Christian?
I don't completely understand what you really sell. This process seems complex. Is this individual for each patient? And where is it done? Is it a special clinic or a machine, or you have to send it to a lab, or how is this working?
So the hospital sends a blood sample to the diagnostic lab, and then in a centralized facility, we do the whole process, and what the doctor receives is a report that gives them a risk. Have to change something now and not wait for the next follow-up.
Okay, so you have your own labs who do that. How can you scale this around the world?
So to scale around the world, we have to partner. In the US, it's LabCorp or Quest Diagnostics. They have the network, they have the lab facilities, and we operate with centralized facilities because the blood can be frozen and shipped.
Thank you. Felix?
In case this works, so I know 6 months earlier that I will have this cancer. How does this change my life? Do I live half a year longer or 2 years, or will I not get cancer?
It's a very good question. Right now, today, with what they have in the hospital is they will start with protective agents for bone. So these are 2 drugs, denosumab or bisphosphonates. The current guidelines only allow them to give these drugs once they have the fracture, once they have the problem. And there is data from literature that it helps to prevent and delay, but you cannot give it just everyone. You really need to figure out the patients that are in dire need for it, and you don't want to give it to patients that don't need it right now.
We have time for one last question.
You have 2 patents. You are the owner?
We are in the process of filing 2 patents. So one is on the bone model, on the next generation device, will be owned by ETH. Then we have another patent that's for creating actually the test result with the fracture risk scoring. So there we want to complete the work at CompagOs, and that's how we split. So the clinical tests are all done in the company.
Thank you very much, Gian. You can go back. Our next entrepreneur on stage will be Bilal Fares from Azure Cell Therapies. Welcome, Bilal.
So let's start with a small exercise. Can everyone please raise their hands and do this? Come on, do it. It's easy, right? Well, in 2010, my grandfather woke up one day and he was unable to do this. And this is how he was diagnosed with Parkinson's disease. I'm Bilal Fares, co-founder and CEO of Azure Cell Therapies, and today it is my pleasure to tell you about our mission, which is to regenerate neurons to defeat Parkinson's disease. And Parkinson is a devastating disease that is on the rise. And this is because so far, it remains without any cure. And you can really think of Parkinson like a fire in the brain, going beyond control, leading to the progressive and irreversible loss of neurons over time. And it is this loss of neurons that leads to the symptoms that the patients suffer from. And so far, all available treatments are symptomatic, meaning that they are similar to throwing green leaves into a burning fire in order to try to make the forest look green again. But today, there is hope, because with advances in stem cell biology and in cell therapy, we can imagine a future where we can stop the fire of Parkinson's and replant neurons into the brains of patients to restore their lives, just as you would replant trees to regenerate a forest after a fire. And the way that cell therapy works for Parkinson is to produce human neurons from stem cells in the lab and to inject these back into the brain of patients exactly where they were lost in order to restore their lives. And no, this is not science fiction, because the first phase 1 clinical trial using this approach was just completed last year, and it already shows signs of clinical benefits in patients, so it works. However, in order for this approach to unlock its full potential, it has to overcome 3 main limitations. And the first is scalability, because it's very complicated, costly, and inefficient to produce these cells. The second is the risk of tumor formation from contaminating proliferating cells. And the third is limited effect sustainability, because we do know that many of the injected neurons will catch the fire of Parkinson's and die. At Azure Cell Therapies, leveraging 18 years of academic research at the University of Geneva, 8 million in grant funding, and a portfolio of 5 patents, we have developed new cell engineering and genetic engineering platforms, which together allow us to develop the next generation of cell therapies for Parkinson's that are scalable, that are engineered to be tumor safe, and that are programmed to put off the fire of Parkinson's disease in the brain, thereby enhancing the long-term sustained efficacy of these therapies. Our team has not only important substantial scientific expertise in this, but also important entrepreneurial experience, and we're supported by an excellent panel of advisors in cell and gene therapy development. And we hope that you would also support us and support our mission to bring the future of medicine for Parkinson's disease. Thank you.
Thank you, Bila. Teresa is already. There is a microphone.
Thanks so much. Are you able yet to predict the outcomes? And a second question is with regards to the tumorigenicity. How do you control the cell growth?
Fantastic questions. So with regard to the outcome, based on the data which is coming out from this initial clinical trial, which is transformative, we know that it's not science fiction anymore, that exogenously produced neurons can integrate and survive and rescue the motor deficits in patients. So we're relying on this. However, what we can also predict is the limitations of applying this. And tumorigenicity is one important one because last year the FDA issued the black box warning for CAR T-cell therapies, which is the highest of them all. But for Parkinson, even if one case of tumor formation happens, then it's game over because Parkinson would not kill the patient in itself. It's a slowly progressive chronic disease, but the tumor will. And so this is why we integrate, this comes to the second point of the question, which is how do we do it? So we have a cell cycle dependent safety switch, meaning that only proliferating cells express a certain protein, which is not toxic in itself, but it serves as a marker that allows us to see that these cells are proliferating. And then we can come with an FDA-approved prodrug, which is brain permeable, which will interact with this protein and lead to the selective elimination of these cells.
A question from Guy.
You mentioned several patents, which is basically a result of 8 years of development. Where they belong, these patents? Where they are located these days?
Great question. So there's 5 patents, 2 on the production procedure, which is scalable, 2 on the safety switch, and then 1 on a neuroprotective protein, which protects the cells. 4 of the patents are actually owned by the Clayton Foundation, which funded this research over the past 18 years. And they have an agreement with the University of Geneva to own the patents and then to basically sub-license them to other companies. And we have an LOI signed already with the Clayton Foundation, and we're negotiating the term sheet for the exclusive license for these patents.
Why they license them to you and not to anybody else?
Great question. So my co-founder is Professor Karl-Heinz Krause, who is the main inventor of all of these patents. He has been working with the foundation over the past 18 years. And what we did is the power of the patents comes all together in combination. And we were able to develop this proposal and this project where we combine this unique combination into this unique product, which is not present on the market today. And the foundation is very supportive of our project. They've been also connecting us with leading foundations in Parkinson research in order to secure additional funding that is needed to drive this project forward.
Thank you very much, Bilal. We're going to stop here with the Q&A. We have 2 more startups before a cup of coffee. Our next entrepreneur on stage is Vincent Forster from Mirai Bioscience.
Thank you. Hi, good morning, everyone. My name is Vincent Forster. I'm a serial biotech entrepreneur and CEO of Mirai Bioscience. Today, I'm excited to show you how Mirai intends to unlock the full potential of genome editing. So throughout history, technological revolutions have reshaped our world. And today, we are really at the genesis of the most transformative wave yet, the convergence of genetic medicine and AI. AI has accelerated the way we do science like unprecedented before, and genetic medicine have given us the key to access many diseases at their roots. At Mirai, we are at the forefront of this revolution, combining the forces of both technology and creating a new generation of genome editing to make disease correction a true reality. Our mission is really to change the status quo here. You can see it will work, but it will clearly not be efficient. It will slip and leave permanent casualty. Our answer is to create novel editors which are tailored and specific to each mutation to unlock new indication and deliver better safety and efficacy. But let's talk about CRISPR for a moment. What you see here in blue is CRISPR-Cas9, the best genome editors today. To correct a mutation, a short sequence called PAM that you see here in green governs the first few steps, which is DNA recognition, binding, and unwinding. So PAM is really important to the targetability, efficacy, and safety of genome editors. Cas9, the original Cas9, is limited due to its PAM, so to increase the targetability and meet more diseases, groups have designed variants which are able to recognize more sequences. But these came at the cost of lower efficacy and lower safety. At Mirai, we're taking a more challenging but more rewarding approach, designing efficient, specific and tailored editors which will deliver better precision and solve the three challenges of targetability, efficacy, and safety. Our Morpheme platform, combining protein evolution, AI, and high-throughput characterization, is probably the most sophisticated CRISPR engineering tool today. Our PAM-specific editors have shown to be able to target new DNA targets with high precision. What you see here in green is how specific our editors are, really decreasing the risk of doing unwanted events on the DNA. We are currently developing cures for rare diseases such as alpha-1 antitrypsin and phenylketonuria. Both are associated with high unmet medical need and high severity. At NIRI, our editors have already shown superior editing efficacy and they really have the potential to be best in class. I'll leave you with this slide and remind you that at NIRI, our vision is really to create a future where genetic diseases are met with permanent hope. Thank you very much.
Merci beaucoup, Vincent. Christian?
Where is the exact status of your program?
So our lead candidate that you saw on the slide, we have created them through directed evolution, and we have tested them on the human mutations which are associated with the disease that I showed you, right? So this is a really key important step toward the translation of these editors. We now have ordered the humanized mice model, and we have secured financial to really move these editors in vivo into this humanized mice, which is a gold standard model, which is really translatable to what will happen in clinic.
Guy?
When I looked at the financial you submitted, you need 15 million with still no turnover. Who is going to fund this money?
So we have a dual business model, if you will, in order to get the financial to be able to run our pipeline. First, we will go to VCs, and VCs, they are investing into non-revenue companies, especially in life science and in pharmaceutical science, but also our platform is able to generate library of these novel, highly precise editors, and we've already been in contact with the leaders in the space, large pharma company, which are willing to in-license our editors. And this in-license deal will come with upfront and milestone payment, which then will de-risk the financials of the financial needs needed to create novel editors.
What is your main challenge you have right now?
Right now, the main challenge is, financing. Of course, we need, as you were saying, a large amount of financing in order to bring this to patients, right? But the main challenge, as in many drug development, is really to show and de-risk each steps on the way and to show that it works, right? And we know that 95% of the drugs are not making it all the way to market, so this is and will remain our biggest challenge all the way through.
Thank you very much, Vincent.
Thank you.
And to wrap up this, the Life Sciences, Biotech and Pharma cluster, Alessia Pacagnella for Drug Bio.
Hi, everyone. Thank you for having me here today. I'm Alessia, the CEO of Drug Bio. Have you ever wondered why the same drugs work wonders for one person but may not work for another? On a more practical level, the same chemotherapy treatment saving one person's life while failing another. Well, the answer lays primarily in a big ecosystem of bacteria living in our bodies, the gut microbiome. This unique ecosystem holds incredible power over how drugs work, but yet pharma still lack tools to understand fully these interactions. On one side, the microbiome can enhance drug efficacy by activating prodrugs, but also by modulating the immune response, which is crucial for cancer therapy. But on the other side, this also leads to critical effects. So 20% of drugs are inactivated by gut microbes, and in 30% of cases, the microbiome metabolism leads to the generation of toxic byproducts. This leads to costly clinical trial failures, wasted resources, and millions of patients not receiving the right treatment. At Drug Bio, we're changing this. So we built the first AI-driven platform that is able to predict drug microbiome interactions from the very beginning of drug development. So using a drug's chemical structure, we're able to analyze the microbial enzymatic interactions and their impact on the bioactivity. But an AI model is only as good as the data behind it. That's why we use a very comprehensive set of data that we build by integrating public curated sources with proprietary wet lab data. We train our AI on this data, and this allows us to unlock new insights for pharma companies that are now able to have an early stage risk assessment. They are also enabled to have an alternative to costly and time-consuming experimental testing. And on the ethical side, they're also able to reduce animal testing. I'm not alone in this journey. Elia, my co-founder and CTO, brings her deep expertise in drug discovery and bioinformatics from Stanford and ETH Zurich. We are part of Medicine, and we've been selected by Partners, which is an incubator from Servier, which provides us with access to labs and expert support. We are currently looking for fundings to expand our team with lab technicians that can help us with data generation. And we are securing pilots with pharma companies to validate our approach. And we are also in open discussion with CROs to integrate our solution. Thanks a lot for the attention.
Christian is ready.
Thank you for the presentation. Do you have evidence that this works?
Yes, so there is increasing evidence nowadays about the interaction between the microbiome and the drugs, and pharma are actually finally realizing that. There are some specific examples like L-Dopa or irinotecan that are drugs for Parkinson and for cancer treatment. So yeah, we are in close contact with Servier that confirms us that they are looking at these interactions.
Thank you. Thank you. Who's going to be the fastest? CellX.
How do you make money?
Okay, so we have a fee-for-service business model. That means that pharma can come to us with a list of compounds, and we give analysis on these compounds, and the price depends on the number of compounds they want to analyze. But we also want to expand doing co-development with pharma companies with royalties, and in the future, in the long run, maybe we would also like to go into drug development ourselves.
Teresa?
The drug microbiome interactions are without doubt very interesting, but the patient microbiome interactions with the drug are even more interesting. How are you going to address that?
So you were asking about like interoperability between different patients. So for now, we place ourselves between in preclinical stage, but for sure that's a very interesting use case that we could maybe tackle in the future. But for now, we want to target preclinical stage, so we identify a profile of the microbiome for specific diseases, and we analyze that.
Thank you very much. Thank you, Alicia.
Thanks a lot.